again, lots of people have thanked Munir and Nathan and Christine, but I'd like to thank them again as well. This is always a great conference, and it's so lovely to be back amongst real people. And for this session, it's so lovely to be amongst young people because I am doing the early career session. So everyone is very, very young in this session, apart from me, obviously. So first speaker is going to be James Yarmolinsky. James is a Cancer Research UK fellow, running a study called Inflammation in Cancer. His background is in epidemiology at Bristol and also a master's at London School of Hygiene and Tropical Medicine, and then spent some time in Brazil, which I'm sure was very exciting too. James's talk is this a short version, James, or is it a longer version of this title? Using Mendelian randomization to evaluate the effect of long-term HMG CoA reductase inhibition on ovarian cancer risk. Right, James, all yours.
Great, thank you. So first, I'd like to thank the organizers of this meeting for inviting me to speak. So I'm going to be sharing some of my work using an approach called Mendelian randomization to evaluate the effect of HMG CoA reductase, which is the drug target of statins on ovarian cancer risk. And I'll also be presenting some recent mechanistic follow-up analysis to this work. So just to provide some context to this analysis, so I'm sure many, if not all of you, are aware already that statins are commonly prescribed LDL cholesterol-lowering medications. And these medications lower LDL cholesterol by inhibiting the enzyme HMG CoA reductase, which is involved in de novo cholesterol synthesis. So there's a wealth of in vitro and in vivo studies suggesting that statins may have anti-neoplastic effects. And importantly, in humans, there are also observational epidemiological studies suggesting that women who take statins have lower rates of ovarian cancer as compared to non-users. However, it can often be unclear whether links between particular exposures and outcomes in observational studies, for example, statins in ovarian cancer, represent causal relationships between these traits. And this is because of the susceptibility of these studies to various forms of bias, which can undermine robust causal inference. So just to give you a sense of the types of biases that can arise in these analyses, so as I'm sure many of you are aware already, in these epidemiological studies, we're usually interested in estimating the effect of a particular exposure X on an outcome Y. So we can think of this, in this case, as statins on ovarian cancer. In these studies, there are often many common causes of both an exposure and outcome, what we call confounders, which can generate spurious associations between our exposure and our outcome. So in practice, we try to adjust for these confounders in our models, but usually our knowledge of confounders is limited to just a subset of the true number of confounders that could be operating. And this leaves a lot of scope for residual confounding in our analyses. Another potential issue is simply misspecifying the causal direction between traits. So in other words, reverse causation. So for example, women with early stage latent ovarian cancer who may have weight loss may subsequently be less likely to be prescribed a statin, which could give the illusion of a protective effect of statins on ovarian cancer. And thirdly, there are often many, many types of selection and information biases in these studies, recall bias just being one of many. So for these reasons, one could argue that conventional epidemiology frequently gets the wrong answer to causal questions. So if we accept this as true, but if we're still interested in examining the effect of a particular drug on a particular disease in humans, and if we can't perform a clinical trial, what is the second best approach available to us? So one potential solution to this is to use an approach called Mendelian randomization, or MR. So in MR, rather than looking at the relationship between a directly measured exposure on a directly measured outcome, we use one or more germline genetic variants strongly associated with an exposure to proxy that exposure to examine the effect of that exposure on an outcome. And we're interested in using germline genetic variants, and I should say that this is similar to what Robert mentioned previously in his talk. We're interested in using these genetic variants as proxies for exposures for a few reasons. So firstly, because germline genetic variants are randomly assorted in meiosis, they should be less prone to conventional issues of confounding that we see in conventional observational studies. Secondly, since germline genetic variants are fixed at conception and cannot be altered by subsequent disease status, these analyses, if performed correctly, should be largely immune to reverse causation. And also with modern genotyping technologies, we're able to measure genotype relatively precisely, which should minimize sources of measurement error. So how does an MR analysis work in practice? So as with any observational study, again, we're interested in looking at the effect of an exposure X on an outcome Y. So for the directly measured traits, the directly measured exposure and outcome, there will almost certainly be some sort of confounding structure in place. The difference is in an MR analysis, we use G, which is our genetic instrument or genetic proxy, to proxy X to estimate the effect of X on Y. And we're able to do this under 3 assumptions. So the first assumption is that our genetic instrument, so again, this could be one or more genetic variants associated with our exposure. The first assumption is that our G is strongly associated with X. So we normally meet this assumption by using genetic variants that are associated with a particular exposure at genome-wide significance. The next assumption is that there's no confounding of the instrument outcome association. So there's no confounding between G and Y. So we wouldn't imagine that most typical conventional confounders, lifestyle, environmental factors would be confounders here because these traits will not alter your germline genotype. The third assumption is that there's no direct effect of our instrument on our outcome. In other words, there's no effect of our instrument on our outcome other than that mediated through our exposure. And we normally examine this through both. We work this into the design of our study, and then we perform various sensitivity analyses to test this. Okay, so returning to the main aims of my study, or our study. So given some preclinical and epidemiological evidence suggesting a protective effect of statins on ovarian cancer, we were interested in examining this using Mendelian randomization and specifically looking at the association between genetically proxied HMG CoA reductase, again, the drug target of statins, and ovarian cancer. So to perform this analysis, we obtained data on around 22,400 women with ovarian cancer and around 41,000 controls from the Ovarian Cancer Association Consortium. And we also obtained data on these women for 5 of the most common histotypes of ovarian cancer. So in addition to these women from the general population, we also obtained data from an independent sample of women with BRCA1 and BRCA2 mutations. So these are women at high elevated lifetime risk of ovarian cancer. So this was obtained from the SIMBA consortium, and this included data on around 4,000 ovarian cancer cases and 31,000 controls. So to develop genetic instruments to proxy HMG CoA reductase, we selected genetic variants that were either in or within proximity to HMG-CR, which is the gene that encodes HMG CoA reductase, that were strongly associated with LDL cholesterol at genome-wide significance, LDL cholesterol being the downstream biomarker of HMG CoA reductase inhibition. So also in secondary analyses, we used a similar approach to construct instruments for 2 other LDL cholesterol lowering targets. So these are the targets for the drug ezetimibe and also for PCSK9 inhibitors. And our reason for doing this is that we were interested in seeing if we were to find a potential protective effect of HMG CoA reductase. We wanted to examine, could this effect, is this effect shared across similar classes of LDL cholesterol lowering medications, or could it be specific to, let's say, the mechanism of HMG CoA reductase? I won't go into too many details here, but we generated causal estimates using inverse variance weighted random effects models, adjusted for some correlation between SNPs, and we performed various sensitivity analyses to examine the robustness of our findings to violations of Mendelian randomization assumptions. Okay, so getting to the results. So these results are scaled to represent drug target inhibition equivalent to a 1 millimole per liter reduction in LDL cholesterol. So when looking at HMG CoA reductase on risk of ovarian cancer, we found around a 40% reduced risk of ovarian cancer. When we looked at this association with our 2 other LDL cholesterol lowering drug targets, so again, the targets for ezetimibe and PCSK9 inhibitors, we found little evidence of association. When we looked at an independent sample of women with BRCA1, BRCA2 mutations, again, we found around a 30% lower risk of ovarian cancer for HMG CoA reductase inhibition. And similarly, we found little evidence of association for the other two drug targets that we examined. So when looking at these findings across our different histotypes, we found the strongest evidence of association for endometrioid carcinoma. But there was little evidence overall for heterogeneity across these subtypes. And these findings also held up in various sensitivity analyses that we performed. Okay, so since after finding this, evidence of a potential protective effect of HMG CoA reductase on ovarian cancer, we were interested in exploring some potential mechanisms that could be governing this effect. So there's recent appreciation that the ovarian surface epithelium and fallopian tube epithelium are likely cells of origin for most ovarian cancers. So given this, we were interested in examining whether an effect of HMG CoA reductase inhibition on ovarian cancer could potentially be mediated through differential gene expression in these two tissues. So to perform this analysis, so we obtained data on, we obtained 103 ovarian surface epithelium samples and 59 fallopian tube epithelium samples from women undergoing salpingo-oophorectomy at Cedars-Sinai Hospital in Los Angeles. And with these samples, we also had germline genotype and RNA sequencing data available. And similar to before, we constructed a genetic instrument, a genetic risk score for HMG CoA reductase using the same approach as before. So selecting SNPs in H and GCR strongly associated with LDL cholesterol. And we performed two analyses. So the first analysis was a transcriptome-wide analysis where we took our genetic risk score proxying HMG CoA reductase, and we examined this for its association with gene expression across around 58,000 genes in both tissues. And then we performed some gene set enrichment analysis, which I'll explain on the next slide. So I want to stress these are still very much preliminary results, but in the initial transcriptome-wide association analyses, we did identify some gene expression signals in relation to our genetic risk score across both tissues. These, however, would not survive a formal FDR correction for multiple testing. And then next, for the gene set enrichment analysis, so how this works is it examines for enrichment of gene expression signals across groups of genes with similar biological annotations or presumed roles in carcinogenesis in relation to our genetic risk score in this analysis. So interestingly, what we found was the TNF-A signaling pathway via NF-kappa beta was the top signaling pathway enriched in both tissues. And TNF-A signaling plays an important role in various pathological processes. So we know that it plays an important role in cell differentiation, proliferation, apoptosis. And also there's some in vitro evidence suggesting an important role of the signaling pathway in ovarian carcinogenesis. And in addition, we also found some evidence for enrichment of some other gene sets involved in different stages of carcinogenesis in both of our tissue samples. Okay. So to conclude, using Mendelian randomization, we found some evidence that for a potential protective effect of HMG CoA reductase inhibition on ovarian cancer risk. This is consistent with some preclinical and epidemiological studies. This finding was replicated in an independent sample of BRCA1 and BRCA2 mutation carriers. We were able to extend the Mendelian randomization paradigm to examining the effect of HMG CoA reductase on tissue-level gene expression, where we found some evidence that, for example, TNF-A signaling via NF-kappa beta may mediate some of this effect in addition to some other pathways. And there are various other ongoing analyses that we're still performing to try to better understand how HMG CoA reductase may be influencing ovarian cancer risk. And yeah, so I'd just like to acknowledge my co-authors on this work in addition to OCAC and SIMBA consortia for providing data for this analysis. I'd like to thank you for listening, and I'm happy to take any questions.